Good morning, everyone. Today, I'd like to talk about CAR-T therapy. Let's define it directly. CAR-T therapy is a specialized form of immunotherapy where we genetically engineer a patient's own T cells to fight cancer. CAR stands for chimeric antigen receptor. By introducing a synthetic receptor into T cells, we give them the ability to recognize specific cancer antigens directly, independent of MHC molecules, and destroy the tumor cells efficiently. Let's look at the mechanism. The process involves 4 key steps. First is the collection. We collect T cells from the patient's blood using a process called leukapheresis. Second is the genetic engineering. In the laboratory, we introduce the CAR gene into the T cells using a viral vector, typically a retrovirus. The structure of this receptor explains why it is called chimeric. It acts as a hybrid, combining 2 essential functions. One is an extracellular receptor that recognizes the cancer antigen. The other is an intercellular signal domain that transmits the signal to activate the T cells. This combination allows the T cell to find and attack cancer cells directly. Third is the expansion. We cannot infuse just a few cells. We need to grow them into millions. To do this, we activate T cells using beads coated with CD3 and CD28 antibodies. This mimics the natural activation signals, stimulating the CAR-T cells to increase rapidly in the culture. Finally, infusion. These engineered and expanded cells are infused back into the patient to hunt down the cancer. Now, let's look at the clinical example. The most successful application of CAR-T therapy so far is in treating acute lymphoblastic leukemia, or ALL, specifically B-cell ALL. In B-cell malignancy, the cancer cells express a specific protein called CD19 on their surface. We designed the CAR-T cells to target these CD19 antigens. The results have been remarkable. Clinical trials showed that in children and young adults with resistant ALL, patients who have had no other options, CAR-T therapy achieved complete remission rates of over 80%. This led to the FDA approval of Kymriah, the first gene therapy available in the US. However, this powerful therapy comes with significant challenges. First is the issue of on-target off-tumor toxicity. Since CD19 is also expressed on normal, healthy B cells, the CAR-T cells destroy them as well. This leads to a condition called B-cell aplasia, leaving patients with low antibody levels. To manage this, patients often require long-term IVIG, intravenous immunoglobulin replacement therapy, to prevent infections. Second is the high cost. Current therapies like Kymriah are very expensive, costing around 30 million yen per treatment. To address this, researchers are developing methods using transposons. This is a non-viral gene transfer technique that is simpler and cheaper than using viral vectors, potentially making the treatment more accessible in the future. In conclusion, CAR-T therapy is a life-saving breakthrough for resistant blood cancers. By lowering cost through new technologies and managing side effects, we aim to make this advantage treatment a standard care for more patients. Thank you for attention.